Good afternoon and welcome to this Chemistry World webinar. I'm Ben Valsler, Chemistry World's digital editor. Now, my role today is just to do a bit of housekeeping before I hand over to my colleague from the RSC, Kathy Page, who's in our policy team. She's going to take you through our topic for today and introduce our guest who we are delighted to have with us. Before I go any further, you will probably have noticed, if you've got the webcam switched on, that I'm joined by Rachel Dance from the Chemistry Deaf Association. She is helping us doing BSL interpretation. If you need her services, then you will find that you can move the webcams around, you can make her camera bigger and so on. So just click around on screen to make the most of Rachel. We're delighted that she's with us and we hope that she's beneficial to you as well. Now, we are using GoToWebinar today, and the best way for you to get the most out of that is to engage with our speakers, ask them questions, take part in the polls and so on. GoToWebinar will be different for everybody depending on what platform you're using, whether you're here on a phone, for example. But the key thing you need to know is that there is a questions box, most likely at the bottom of the GoToWebinar panel or on the side of the screen if you're on a phone. Get your questions in at any point throughout the presentation. We have 3 excellent speakers. We should have 15-20 minutes for questions at the end. So don't leave your questions till the end. Anything you would like to ask at any point, get them in as soon as you can. And that's really the best way to make sure that, that this is not just a broadcast, but actually a webinar, a proper engaged event itself. So do please ask those questions. It really helps us to make the most of it. We will also be asking you a survey on exit. Again, that helps us to shape future webinars and to know what we did right this time and make sure that we do more of that in future. So do please take a couple of minutes to fill in the survey. Now, speaking of after the webinar, if you find that you missed any bits or if you'd like to come back and watch any more of it, then we will be making this available as a recording. Everybody who registered will get sent a link to the recording in the next few days. And for those of you who are with us live now, you will also get a certificate of participation. That's just our way of saying thank you for joining us for the live event to really make it truly engaged. That's basically all I have to say for now. So I'm going to hand over to Kathy Page. Again, she's from the RSC's policy team. She's worked with us here at Chemistry World to put together a series of webinars looking at progressive plastics, answering key questions. And this time we're looking at how polluting plastic really is. Kathy, thank you for joining us. Take it away.
Thanks so much, Ben. Hello, everyone, and thank you for joining us for our first joint webinar with the Microbiology Society. It's sometimes a bit more difficult to get a sense of the audience virtually, but we're really happy to see over 1,000 people have signed up for this webinar from all over the world, from the UK to Slovenia, Nigeria, the Philippines, Brazil, Canada, and many more. So welcome, everyone. My name is Kathy Page, and as Ben mentioned, I am a Programme Manager in the Science Policy Unit here at the Royal Society of Chemistry. For those who don't know, the Royal Society of Chemistry represents chemists in the UK and worldwide, with over 45,000 members across academia, industry, government and other sectors. And our policy team works to make sure the best science in chemistry is available for decision makers. Today, we're delighted to be presenting this webinar in collaboration with the Microbiology Society, a membership charity for scientists interested in microbes, their effects and their practical uses. It is one of the largest microbiology societies in Europe, with a worldwide membership based in universities, industry, hospitals, research institutes and schools. So today we're thinking about plastic pollution, its chemical and microbial impacts, but also how we can harness the power of nature to deal with discarded plastic. We know that plastics provide many benefits to society, but our throwaway model of consumption has led to vast amounts of pollution in our natural environment. This can come in different forms, from large localized patches of rubbish or widely dispersed microplastics barely visible to the human eye. Chemists and microbiologists, along with other scientists, study this pollution, its effects and possible solutions. The RSC has been publishing and promoting policy and research around plastics for a long time. You might want to check out our previous webinars and current plastics explainers online. Those cover the topics of biodegradability and degradation of plastics, the future of plastics recycling as well. Our website also has our policy positions, extensive journals, and our reports on science to enable sustainable plastics. Our new explainers looking at plastics in the environment will be coming out at the end of this year, so look out for those. And we also have a new podcast all about plastics. If you search Brought to you by Chemistry on YouTube or your podcast app, you should be able to find that. The Microbiology Society's recent project, A Sustainable Future, looked at how microbiology can help achieve the UN Sustainable Development Goals. Their report, Transitioning to a Circular Economy, Opportunities and Challenges for Microbiology Research and Innovation, along with some case studies, podcasts and other resources available on their website. You can also find a new climate change hub on there with information about how microbes can play a part in sustainable solutions. So today we are having this webinar to hear from some experts about plastics impact and hopefully answer a lot of your questions. With that in mind, as Ben said, we really want to hear from you. So please do ask those questions at any point, and we'll hopefully have lots of time to discuss those with our speakers in the second half of today's webinar. And we've got three really excellent speakers for you today. Coming up a bit later, we've got Dr. Robin Wright, who is a postdoctoral fellow at Dalhousie University, and Dr. Jesus Ojeda Ledo, associate professor at Swansea University. But first, Dr. Tanya Narencic, who is an assistant professor at the School of Biomolecular and Biomedical Science at University College Dublin. Her research focuses on elucidating the metabolism of bacteria and using this information and synthetic biology tools to exploit bacteria to make high-value products. So, without much further ado, over to you, Tanya.
Thank you very much, Kathy. And okay, that should be the one. Perfect. Well, thank you very much for joining us today. And what I will be talking, oh, sorry, it's actually showing the wrong screen. Perfect. We're going. Thank you very much for joining us today. I will be talking today about how can we employ and exploit microorganisms to offer a solution to plastic waste. And I would think that most of you would be aware of the issue that we're dealing with, you know, the plastic, how much we're producing and how much of plastic waste has been accumulated. And having that in mind, I would like to ask you, what is your opinion? Should we just ban all plastic? So you should get a poll now to pop up, and please let me know just a simple yes or no. What do you think? Should we ban it or not?
So you should be able to see on your screen now the poll there. If you just clicked one of those two options there, we will be getting the results on the screen in just a short amount of time. Do remember you can ask questions as well if any questions come up when you're thinking about your answer here. And I'll just give you another 5 seconds. 5, 4, 3, 2, 1. Okay, could we have the results, please? Very interesting. Okay, back to you, Tanya.
I'm actually delighted to see this result, and I'm hoping to chat to you more about you know, why is this the case? Why do you propose not to ban, majority not to ban? So we are then going to continue. I promise I will tell you some things. I'm not just going to ask you questions, but before I tell you things, I would like to ask you one more question. So, okay, we do produce a lot of plastic. We most probably do that because we need it a lot. And there is one way of dealing with plastic when it finishes its life, and that is recycling. But what I would like to ask you is, what do you think how efficient recycling is, in terms of what do you think how much of plastic that we produce annually we would recycle? Again, you will get 3 options now. Do you think it's about 30%, about 50%, or higher than 50%?
Brilliant, thanks Tanya. You should be able to see that on your screen again. So roughly how much plastic do we recycle? You've got 3 options this time. So have a little think about which one you think is correct, and I'm sure we'll find the answer straight away afterwards. I'll just give you a bit of time to have a look at those and have a think about what your answer is going to be. I can see we've already got some great questions coming in into the Q&A box, so do keep those coming in. And I'm going to give you another 5 seconds. seconds. 5, 4, 3, 2, 1. Brilliant.
Brilliant. Thank you very much for this. Around 30%. Again, 80% majority is correct, and that is great. Okay, so I will then continue with my presentation. So the reason why I wanted to raise this question is, yes, we in the EU, and again, this number will be different depending on the country, member country of the European Union. We do have more accurate, say, data, but when I was looking for the data for the States, for example, I couldn't find a number that would be kind of more exact. So in the EU, approximately 30% of plastic is collected and sent down recycling route. However, I'll tell you a devastating number here, and that is that only 4% of what is recycled is being reused. So there's a massive gap between what we recycle or what goes down recycling route and how much of that recycled is being used again. The main issue there is that we're producing something of the lower value than the starting material. So virgin material is of higher value than recyclate. So what we're proposing to do is, rather than recycle plastic, to upcycle it. What would that mean? So turn this plastic waste into something that's of higher value. How do we do that? So this whole thing started in the 90s when Kevin O'Connor, who was my supervisor while I was doing my postdoc, came up with an idea to develop a bioprocess in which he would use bacteria, and bacteria would be fed by plastic monomers. So in the first phase of the process, he would take plastic, and you can see here polystyrene cup, polyethylene terephthalate bottle, or polyethylene bag. And in the first phase, it was a chemical step. Say in pyrolysis, this plastic would be converted into monomer components. And then these monomer components were used as a feedstock to feed our bacteria that would then produce PHA. So before I continue on what PHA is, I will just tell you that now we know that some of these polymers, such as polyethylene terephthalate, for example, is hydrolyzable polymer. So there are enzymatic routes to degrade polyethylene terephthalate into its monomer constituents, which are here shown, ethylene glycol and terephthalic acid. And then again, we can use this to feed our bacteria. But why would we be interested in producing polyhydroxyalkanoates? So polyhydroxyalkanoates are biological esters, polyesters, that are naturally accumulated by a lot of bacteria. Similarly to us, the way we store glycogen, for example, as carbon and energy source, bacteria store these polyhydroxyalkanoates. So it's a storage polymer made out of R3-hydroxyalkanoic acids, and you can see here what they look like. And there are over 150 monomers that can make these polyhydroxyalkanoates. So it's not one polymer, rather a family of polymers similar to plastic. And again, similarly to plastic, these different polyhydroxyalkanoates will have different characteristics. So here you can see a monomer of polyhydroxybutyrate, which is a representative of short-chain polyhydroxyalkanoates. Why are they called short-chain? Well, you can see this group here, it's only a methyl group. And these polymers would be more brittle. And then this methyl group can be replaced by an ethyl group, or the chain can grow up to 14 carbons in total. And the longer the chains are, these polyhydroxyalkanoates will have slightly different characteristics. They will become more oily, say. So if you have short chain length, you will have a brittle polymer when you extract it out of the bacteria. And if you have more of medium chain length hydroxyalkanoic acids as monomers, you will get more oilier polymer. And you can see here these lovely bacteria with these blobs that look like lipids inside, and that's polyhydroxyalkanoate in the bacteria. We're interested in polyhydroxyalkanoates because they're elastomeric and thermoplastic. So when we extract them from bacteria, you get something that looks like plastic. They're also biocompatible, but the key word here is that they are biodegradable, because they're bacterial carbon and energy storage. Bacteria have to have a capacity to tap into this storage and, you know, get carbon and energy when they need it. So bacteria have the capacity to degrade these polymers. So going back to our original kind of story, so polyethylene terephthalate is a hydrolyzable polymer, and there are plenty of enzymes now discovered and shown to be able to degrade polyethylene terephthalate and produce some dimers as well, but these are the main products, the end products of enzymatic hydrolysis of polyethylene terephthalate, so ethylene glycol and terephthalic acid. And this is where a key player comes in. So a very powerful bacterium called Pseudomonas umsongensis G16 that we have isolated and identified. And we found out that this bacteria can consume both ethylene glycol and terephthalic acid as carbon substrates. And we have identified a genetic blueprint here. You can see some of the genes that encode for enzymes that are involved in metabolism of ethylene glycol and terephthalic acid. So because Pseudomonas umsongensis can grow on ethylene glycol, but not so great, we have then suscepted this bacterium to adaptive laboratory evolution, in which we were offering this bug increasing concentrations of ethylene glycol, and we were selecting for fast growers here. And by that, we were able to get an evolved strain that now was able to use both ethylene glycol and terephthalic acid at the same time and grow well. You can see here with this orange kind of curve, and accumulate PHA. So why was this important? As I said, you can use enzymes in the first step to degrade polyethylene terephthalate, so enzymatically, and get a mixture of ethylene glycol and terephthalic acid, and then you can use that mixture directly and give them to bacteria that then can use this as a carbon and energy source and produce something that's valuable. So by doing this, we come up with a concept that we call open up cycling loop. So what happens there? As I explained, you have PET as a starting material, you enzymatically degrade it and get ethylene glycol and terephthalic acid, and you've seen that we can produce PHA. But what we also were able to do is incorporate a synthetic pathway in this bug. And now Pseudomonas umsongensis not only can produce PHA, but it can produce hydroxyalkanoic acids that can, or we then use these alkanoic acids in chemical polymerization, and we have made a novel biopolyurethane, another type of plastic. So we have made a bio-based, novel type of bio-based plastic. And what do we need to do to move from just proof of concept into something that's realistically applicable? Well, as I said, we still have a two-phase process. So if we look at the first phase, which is polyethylene terephthalate conversion into ethylene glycol and terephthalic acid, last year a seminal paper, and you can see here the title of the paper came out in Science, if I remember correctly, and they have actually shown that this enzymatic hydrolysis is applicable on an industrial level, and it's a very efficient process. What the authors of this paper shown was that you can then extract ethylene glycol and terephthalic acid from this hydrolysate and make not virgin, but you can remake polyethylene terephthalate. What we're proposing to do here, as I said in previous slide, to directly use what you get from enzymatic hydrolysis to feed bacteria, and thereby kind of circumvent the quite expensive process of extraction of ethylene glycol and terephthalic acid. Now, what else do we need to do to improve the process? Well, again, look at the metabolism of bacteria and how quickly, how efficiently they utilize ethylene glycol and terephthalic acid. Ethylene glycol is an alcohol, it's a diol, it's a short molecule. Most bacteria, if they have a pathway to metabolize ethylene glycol, they use it just as energy. But what we need is that ethylene glycol becomes an efficient substrate. And again, you have two seminal papers that came out of Greg Beckham's lab and Nick Work's lab, and they have engineered bacteria for very efficient utilization of ethylene glycol. And now it's a different Pseudomonas, again, very powerful Pseudomonas KT2440, can now grow with half a molar and 1 molar ethylene glycol. So what am I doing? Well, I'm focusing on terephthalic acid metabolism. So terephthalic acid is an aromatic hydrocarbon, and don't be surprised, Pseudomonas are amazing organisms that have the capacity to metabolize a lot of aromatic hydrocarbons. However, terephthalic acid metabolism is not found frequently enough in nature, but our GO16 has the capacity to metabolize terephthalic acid. And this is the first part of this metabolic route, where terephthalic acid, by the activity of dioxygenase, dehydrogenase, and reductase enzyme, is converted into protocatechase. And protocatechase further is cleaved and converted into key intermediates, which are acetyl-CoA and succinate, which then enter the TCA cycle or citric acid cycle and provide energy. So we have identified the genetic complement that allows Pseudomonas umsongensis to grow with terephthalic acid, and we were functionally characterizing it. And we have then placed this operon into aforementioned KT2440. And what happened was KT2440, that didn't have the capacity to grow with terephthalic acid before, now can grow with terephthalic acid. But what we have noticed is that if you look at the biomass of GO16 here and compare it to the biomass of KT2440 equipped with the same genetic complement, KT2440 cannot achieve about half of biomass that GO16 can, and very low amounts of PHA. So we then started looking more closely into the metabolism of GO16, and we have noticed that GO16 has essentially two sets of pathways for the metabolism of protocatechase, and we're now trying to learn more to tune better this part of the pathway and improve terephthalic acid metabolism in GO16, thereby removing certain obstacles that exist in terephthalic acid metabolism. And we're also learning from that and trying to improve metabolism of terephthalic acid in KT2440. Because I don't have too much time to talk about this, if anyone is interested in why would we have two Pseudomonas strains now growing with terephthalic acid, I'd be happy to discuss that with you later. Another thing that GO16 is absolutely amazing in is that I have mentioned that PHAs, you have kind of two classes of PHAs. We have short chain length PHAs, and I mentioned polyhydroxybutyrate as a representative, and then we have medium chain length PHAs that are mainly accumulated by Pseudomonas species. So you would have Pseudomonas species that can accumulate medium chain lengths. A few of them would have the capacity to accumulate polyhydroxybutyrate, but it is very rarely found in nature that a bacterium can accumulate both natively. And our GO16 can do that. So we have also identified the blueprint that allows GO16 to do this. And we have identified then, we looked at different carbon substrates, such as gluconate, glycerol, terephthalic acid, ethylene glycol, and many others. And we have shown that depending on the choice of the substrate, you can tailor the composition of PHA. And that is very important. I have also mentioned that polyhydroxybutyrate is very brittle, and I don't have it here, but it looks like a polyethylene bag, but when you apply a little bit of force, you can rip it easily. And then I did mention that medium chain length PHAs, they can be more rubbery, and the longer the chain is, they become more oily. So you can imagine if we can kind of find a satisfying composition of these monomers, and we create some, let's say, blend between these monomers, we can create materials that have specific properties, and we can then push them down towards specific products. And there's another very important feature that relates to this monomer composition. I did also say when I was defining polyhydroxyalkanoates that they are biodegradable materials, and they are. We know that there are enzymes that can depolymerize these materials and completely degrade them, convert them, if we're talking about an aerobic process, into water, biomass, and carbon dioxide, or if we're talking about an anaerobic process, you will not have carbon dioxide, you will have methane. But then when we tested in a big study, when we tested different materials, including our short chain length PHA polyhydroxybutyrate and a representative of a medium chain length polyhydroxyoctanoate, we have tested their biodegradability in different environments. So we just extracted the polymer and we simulated the environments. And you can see here their behavior in soil. So over 300 days, we were looking at biodegradation, and polyhydroxybutyrate, represented in green here, completely degrades within 120 days. The mixture of the two polymers behaves similar to PHB. However, PHO, when it's just left like that in soil, it looks like it's not really degrading well. Similar happened in fresh water. While PHO was degrading better, it still wasn't as good as PHB. And similar happened in marine environment. So if we were to make a product from PHA, even though that material is biodegradable on its own, we cannot just simply throw out that product in the environment and expect that it is going to disappear. There are, again, other things that are related to how biodegradable something that we call biodegradable is, and if you have questions about that, I'll be happy to talk about it. But good news is that, if you now take these biodegradable plastics and manage them post-use, you have way more options than just for a conventional plastic. I mentioned recycling. We know incineration is also a big thing for plastic. But with bioplastic, we have some types of bioplastic that we can compost at home. Polyhydroxybutyrate, again, here is compostable at home. Polyhydroxyoctanoate, while it degrades better than in soil, it still is not great as home composting. In anaerobic digestion, again, completely novel option for biodegradable plastic. Polyhydroxybutyrate performs really well. Polyhydroxyoctanoate, not so great. But industrial composting, both materials, and for that fact, many other materials that we have tested, they all show great performance in industrial composting. So essentially what I wanted to tell you here is that we're doing some great research trying to solve a plastic waste issue by opening up novel possibilities and producing something that's very valuable and has different type of management options compared to regular plastic. And by this, I would like to finish and just say thank you first to you, and then many people that were instrumental in getting these results. And obviously funding agencies, primarily Horizon 2020, who has funded 3 projects that were focusing on developing bioprocesses related to bacteria to convert plastic into biodegradable plastic and other valuable products. Thank you very much.
Thank you so much, Tanya. That was fascinating, really interesting there. And if you could stick around, we'll have some questions for you a little bit later as well. Next up, we have Dr. Robin Wright. As I mentioned, Robin is a postdoctoral fellow in Morgan Langell's group at Dalhousie University in Nova Scotia. Her recent projects have included looking at COVID-19, the soil microbiome, as well as some plastosphere metagenome studies. And it's the plastosphere she's going to talk to us about today. So take it away, Robin. There we go.
Thanks, Kathy. Can everyone see that okay? Yeah, I'm going to assume so. Hi everyone. So first of all, thanks so much to the Microbiology Society and the Royal Society of Chemistry for asking me to speak here today. I'm going to tell you a little bit about biodegradation and microbial community dynamics within the PET plastosphere. So as I'm sure that you're all aware, the oceans are now full of plastics. And once in the oceans, these plastics experience a range of different processes. For example, they can be transported long distances by wind and currents. They can be fragmented by wave action. They undergo weathering through processes such as photodegradation by the sun's UV rays. They can be ingested by a whole host of organisms, and also they can be colonized and potentially biodegraded by microbes. The microbes that colonize plastics in the oceans are an often distinct group of microbes that are known as the plastosphere, a microbiome for plastics, if you will. So this biofilm colonizing plastics in the oceans is made up of a diverse mixture of bacteria, fungi, and even some larvae of larger organisms. But we don't really know definitively what the metabolic capacities of these microbes are, or more simply, what they're doing there. It's been suggested that they're just surfing on the plastics, or they're just kind of being taken along for the ride, that they're pathogenic and they may be traveling larger distances on the plastics than they would otherwise, and also that they could be degrading the plastics. So plastic biodegradation, as we've kind of just heard a bit about, it refers to the biological breakdown of plastics to smaller intermediates and eventually to carbon dioxide and water. And the biggest question that we have in the case of marine plastic degradation is not necessarily just how plastics might be biodegraded, but can they even be degraded in the marine environment? And I've chosen to focus this question on PET, or polyethylene terephthalate, the plastic often used for producing things like this plastic bottle. And as you've just been hearing a little bit about, that's mainly because in the case of PET, there's several examples of bacteria that are capable of degrading the PET, and the best known is probably that of Ideonella sakaiensis, which is a soil bacterium that was discovered a few years ago that's capable of degrading PET. And it's not just knowing that this degradation is possible, but also knowing how it's carried out helps us to be able to look for this occurring in other environments. So this bacterium first secretes a PETase, an esterase, or an enzyme that breaks down the long chains of PET into their subunits of MET or BET, and then it uses a metase, a second fairly similar enzyme, to further break down this MET or BET into terephthalic acid and ethylene glycol, both of which, as you've just heard, are known to be quite readily degraded by a range of different bacteria. And so since this discovery of the Ideonella sakaiensis and the PETase, homologs of the PETase have been found in marine metagenomes, so in DNA sequences taken from the oceans, but there hadn't really been any direct confirmation that this is actually happening, or that marine bacteria are carrying this out. So as part of my PhD, I set out to investigate marine PET biodegradation. And to do this, I went to Porthcawl Beach in Wales. I collected some plastics, and then I removed the biofilm from these plastics, and I used these microbes, so ones that were already colonizing plastics, for a couple of different experiments. First of all, I added them to water, nutrients, and one of a few different types of PET, and I examined how this community of microbes changed across 6 weeks of incubation, taking samples on days 1, 3, 7, 14, 21, 30, and 42. And I used these samples to both sequence the microbial community and to look for breakdown products of PET in the surrounding water. And then as a second objective, I used the same setup to try to isolate some bacteria that might be able to degrade PET. And in both parts of this, I used both PET and also BET, which can be both a breakdown product and a production intermediate of PET. And while I did use a few different types of PET, what I'll be focusing on today is low crystallinity PET. And so the crystallinity just refers to how tightly packed the chains of the PET are. So in the case of the low crystallinity PET, more of the chains are quite loosely packed, which just makes them a bit more accessible for microbial attack. So how does the plastosphere change across 6 weeks of incubation with PET? Well, in this graph, each point represents an individual microbial community, with points that are closer together being from communities that are more similar to one another. And the points get darker as we go from 1 day of incubation to 6 weeks of incubation. So at the top here, this is our community that we took from the plastics collected from the beach. Over here, we have the microbial community growing with BET, so the PET monomer or the breakdown product. Here we have the community growing with low crystallinity PET. And then here we have the communities that were growing with other types of PET. So this just shows us that our communities are different depending on the type of PET that they're grown with, and that they're all different from the starting community that we initially took from unidentified plastics. But what we were really interested in is which microbes are driving these differences. So here this heat map has each section being for a different treatment, each across the 42 days or 6 weeks of incubation. So each column is from samples taken on a certain day. And then over here on the right, I've just got a blob representing the maximum abundance of each taxa and the taxonomic classification. And now I'm just showing the BET and also the low crystallinity or the amorphous PET biofilm samples. And what we can see here is that for BET, the taxa that are driving the differences are generally abundant at the early incubation stages. So that's these brighter yellow colors showing that this taxa, for example, is most abundant on day 7. And what we see is that for the low crystallinity PET, they're tending to be abundant at later time points. And that's probably just because it takes longer for them to even try to degrade the PET than it does for the BET. And I also just want to highlight that some of the taxa that we're seeing here are within some of the hydrocarbonoclastic groups. So these are groups of bacteria that preferentially like to grow using hydrocarbons such as oil or constituents of oil. And it's the presence of these taxa that usually leads people to suggest that plastic biodegradation might be occurring. And these are actually making up a maximum of 15 or 25% of the communities. So we can see that the community changes over time and that some of these changes might be due to taxa that could degrade plastics. But this is not proven degradation by any stretch. So from the isolations that I mentioned previously that used PET and BET, we managed to get 2 bacterial isolates that seem to be able to grow with these substrates as the only sources of carbon, which we identified as Thioclava and Bacillus species. And first of all, we just had a quick look at whether DNA sequences matching these isolates were present in our communities. So that's the ones that we incubated over 6 weeks, shown at the top. So for the case of Thioclava, our isolate was most abundant in the inoculum, but we also detect sequences matching the isolate in previously published plastosphere studies. And it was most abundant in samples from around the UK and the Mediterranean Sea. We also find Bacillus in some of our community samples, although we typically find it to be of really low abundance. And we only really find it to be abundant in 1 day, so day 7 of our amorphous or low crystallinity PET samples. And we also found that in the other plastosphere studies, it was really only present in European plastic samples, but in one of these, it did make up 25% of the community. So having identified some potentially degrading candidates and satisfying ourselves that they were present in plastosphere samples, we wanted to know how they were degrading PET. So we incubated each of our bacteria as well as the known PET degrader Ideonella saccharensis with PET, BET, terephthalic acid, and also fructose as a positive control. And then we performed metabolomics on all 3 bacteria, as well as exo and cellular proteomics on our 2 isolates. So the idea was to detect degradation products and intermediates using metabolomics, and then the proteins or enzymes that each of our 2 isolates was using for degradation using proteomics. So through this combined approach, we were able to get this overall picture of PET degradation. And what we were able to confirm, and what we see here now in green, is that we detect the expected intermediates of PET degradation with each of our bacteria. So Thioclava, Bacillus, Ideonella, and also our community. So when we incubate with PET, we detect BET. When we incubate with BET, we detect MET and terephthalic acid, and so on. And we also have a couple of extra intermediates that we're not entirely sure on the pathway for their degradation. But we also find that for the case of Thioclava, we have enzymes that are very similar to those used by Ideonella for each of these steps, and that these are more abundant when we're incubating with the PET or the BET, for example, than when we incubate with the fructose. So we have an enzyme similar to the PETase and also one similar to the METase, as well as enzymes for all of the downstream steps necessary to actually use both the terephthalic acid and the ethylene glycol for growth. However, in the case of Bacillus, there were no enzymes in the genome or the proteome that were homologous to those used for PET degradation in our known terrestrial PET degrader, Ideonella. But what we do find is a really huge number of oxidases and monooxygenases that are all far more abundant when Bacillus is incubated with the PET substrates than when it was incubated with our fructose control. So this kind of led us to hypothesize that perhaps rather than this pathway that both Ideonella and Thioclava use, maybe Bacillus just degrades PET through a loss of really non-specific oxidation. So we took a look at the pieces of plastics after they'd been incubated with the isolates and our community, and we used FTIR spectroscopy for this. So very simply, this allows us to look at changes in different chemical bonds or in the structure of the plastics. So for example, an increase in this ratio indicates that we have an increase in the number of carboxylic acid, carbon-oxygen double bonds on the plastic surface, which is indicative of hydrolysis or breakage of the PET chains. So what we see is that we have increases in the surface oxidation for PET that was incubated with both Thioclava and Bacillus, in blue for the Thioclava and red for the Bacillus respectively. But the much larger increase in oxidation that we see for Bacillus is in accordance with the non-specific oxidation that we suggest is probably taking place. And you might notice here that with the community, which I've shown in yellow, some of these ratios are increased relative to the controls with no inoculum, which is this grey here, but none of these increases are statistically significant. So obviously it is possible that we're not seeing any degradation by the communities, but we also think it's possible that because we're looking at a whole microbial community, any additional monomers or N groups that are generated might just be being used up by other community members really rapidly. And that is just a hypothesis, but we do think it is possible that degradation is more efficient by communities than by isolates. So just to quickly summarize what I hopefully have shown you here, isolates from the marine plastosphere can degrade plastics. Thioclava uses the same enzymes as a terrestrial bacteria does, while Bacillus seems to use a previously uncharacterized pathway. A community can also degrade plastics, but we do need to do some more work to confirm the mechanisms that are used, and hopefully also to do a little bit more work into looking at the pathway that Bacillus seems to use for this. And with that, I would just like to thank everyone that I was working with, both in the UK and since I've come to Canada, and also my funders for some of these parts. And thank you to all of you for listening. I think we'll be taking questions at the end of the session, but please do also feel free to reach out to me if you have any questions. Thanks.
Thank you so much, Robin, and we look forward to seeing you again in a little bit for some questions. Thank you. So finally, we're delighted to introduce Dr. Jesus Ojo de Ledo, Associate Professor at the Faculty for Science and Engineering at Swansea University. Jesus' research focuses on plastic and microplastic analysis, plastic degradation, and the sampling, identification, and quantification of plastics and microplastics in complex environmental matrices, often using spectroscopic tools. Thank you so much for joining us, Jesus. Over to you.
Thank you. So I hope you can see my screen now.
Yeah, looks perfect.
Thank you. Well, I'm going to talk a little bit briefly about methodologies for sampling and analyzing microplastics in the environment. So microplastics are defined as any solid particle or rubber that is between 5 millimeters and 1 micrometer in size. That includes fibers as well. And nanoplastics are those below 1 micron in size. Where do they come from? Well, they normally come, what we call the primary microplastics are those that are released into the environment already as small microparticles. They could be, for example, from tires, from car tires, or from our shoes, or from washing the synthetic fibers from our clothes, or also from cosmetic products that were including microbeads as exfoliating agents. They have been banned already, but they were already being a source of primary microplastics into the environment. But you can see that the vast majority is what is called secondary microplastics, and those are resulting, for example, from rests of fishing nets, or the degradation of bigger plastic as waste in rivers or in the ocean. So the bigger plastics, due to erosion, abrasion, or photodegradation, they break down into smaller and smaller particles, and they then become microplastics. So we have a challenge to detect, to see how many particles are in the environment, and what are the kind of polymers that could be potentially polluting our environment and ourselves, or affecting our health and the health of any living organism in there. So despite the difficulty of detecting those microplastics, it's very important to investigate their presence, and not also the presence of what is the fate of those pollutants, what is the impact that it could have for the living microorganisms, for the living organisms and the ecosystem. So there have been many studies to collect and to analyze microplastics, but some of those studies are very difficult to compare. while there is expected variability in terms of sources of releases, locations, different populations, there are also some differences because of sampling methodologies and analysis protocols. So although we are seeing more and more consistent methodology, there is still lack of consensus, and there is no standard operating protocol currently for the analysis of this pollutant. And that is the challenge, to see if we can harmonize the better protocols for sampling, collection, pre-treatment, and analysis. What about sampling? It depends on what we are going to sample and what are the expected sizes in there as well. In case, for example, of surface water, the most common way of capturing microplastics is the use of what is called manta nets or Newton nets, plankton nets. So you have a net in a boat, or you can fix them, for example, in a river, and then you collect plastics from the water. If you know the flow rate and the time of the sampling, you could then capture and know the amount of particles as a function of the amount of water that has been passed through the net. There is a limitation of the size of the mesh. So normally the smallest size is about 300 micrometers, or something smaller than that will be difficult to capture in this kind of devices. When we have fresh water samples, we can have what is called a subsequent filtration system. So you can have in water pipes, or if you have a submergible pump, you can put it in a water tank or in a small lake, and pump water and pass them through a series of filters where you can capture also particles at different sizes. And for example, groundwater or freshwater sediments, unfortunately, there is still significant less data for these systems, and it's a technique that is a little bit more specialized and requires more complex sampling methodologies. So far, the use of HES benthic samplers or multi-level groundwater monitoring systems installed in boreholes have allowed to collect groundwater and then the capture of microplastics. This is an example and some more photos of how the Newton nets and manta nets look like for the sampling of surface waters or rivers or larger lakes. And this is some examples of the subsequent filtration. As I mentioned before, the limit of detection will depend on the size of the mesh that you will use. For the case of nanoplastics, using some special membranes, some sampling methodologies have been reported with membranes of a few 100 nanometers. Having said that, that unfortunately is not applicable everywhere, because when we're dealing with nanoplastic, we might need to have a very fine membrane pore size, and that will create problems of pressure or clogging. For example, if we're going to be sampling in wastewater treatment plants, that might not be possible. What are we going to do about it? Are we doing something to harmonize these techniques? Well, yes. From last year, there was a working group established in ISO, which is called the Working Group 16 in the Committee for Water Quality. And particularly for the subcommittee 6, the sampling, for the development of a more standardized approach and to generate a standard for sampling microplastics in water. So that is a work in progress to try to harmonize different protocols. So now that we have capture, or we have the sampling of microplastics, we might need to pre-treat them in to analyze what we want, to identify the polymers, or to count the particles, or to know the mass. On occasion, some of those samples might be covered in fats or oil. For example, from washing machines, you will have threads of soap or fats or dirt. Some microplastics might be colonized by microorganisms. If you are sampling wastewater, you will have plenty of biogenic organic matter. Or also, when you are capturing those fragments in nets, you will get also sand, or you will get other suspended solids that are not plastic. So we might need to separate and to pre-treat the microplastic to be able to count them later. One of the possibilities is separate them by density. This is an example of a sampling device, of a density separation device designed by the University of Exeter the Plymouth Marine Laboratory, where you can, using different densities, make the plastics float. So polyethylene and polypropylene, for example, float in water. But if you put water, for example, saturated sodium chloride, you will get different flotations, and therefore you will be able to separate the plastics from other sediments. On occasions, your problem would be the presence of organic matter. In those cases, you might need to apply some kind of oxidative treatment, either with enzymatic digestion or by using, for example, hydrogen peroxide. This is an example of samples taken from wastewater, and you can digest the organic matter and get the microplastics to be analyzed. If there is much more organic matter, for example, from aerobic or anaerobic treatment stages in wastewater, we might need to have a more aggressive approach, and for those, the Fenton reagent has been applied. We have demonstrated that the use of Fenton reagent for the pre-treatment of wastewater treatment plants do not affect significantly the infrared spectrum of the microplastics, and they are still able to be analyzed. I don't know if that's a good thing or a bad thing, because that means that we can still count them, we can still identify them, but that also means that they are very resistant. So once the microplastics have been isolated, they must be analyzed for quantification and identification, and that's usually achieved by spectroscopic or microscopic techniques. The most common one is visual sorting using optical microscopy. So just with the help of a microscope, you will be able to discern, to pick up, to identify, to see the microparticles. That could have some problems. It might be a little bit unreliable in the because some of the particles that could look like microplastic might not be a plastic, or some that don't look like a microplastic could be a plastic and might be left out. There are some approaches, for example, staining the samples with Nile red, and that will allow you to identify a little bit better what are polymer, what are those samples are from polymeric nature, and you can then discern them. You could also use scanning electron microscopy, which will give you a much more detailed analysis of the surface of the material. It will allow you to also analyze nanoplastics, which you won't be able to see with an optical microscope. You will get very high resolution images, and you could also have elemental analysis. However, obviously, the availability of instruments and the expertise of an operator make this approach a little bit costly. We could also analyze them using pyrolysis, so pyrolyzing the plastic and then analyzed the compounds using separate them in gas chromatography and then detect them with mass spectrometry. It's a very sensitive technique, so it also allows you to analyze nanoplastics, but is destructive. It has been reported recently the use of pyrolysis GC time of flight mass spectrometry for the analysis of nanoplastics. It will allow you to have the identification of the polymer type, and it could also give you information of the additives in the plastics. But because it's a destructive technique, you won't be able to know the number or the size or the morphology of your material. The most common technique is infrared spectroscopy. It's one of the most common and established techniques for the analysis of polymers. So the spectrum of different polymers is already known with infrared spectroscopy. Therefore, you can identify the polymer type by analyzing the different functional groups in their organic structure. And with the use of micro FTIR, you can also have the size and the morphology of the plastic, and you can count them. For example, in there you can see a photo of a filter membrane with different microplastics, and based on the spectrum that they have, you can also have the identity of those polymers. Similarly, you have Raman is also a vibrational technique that with a laser you can get from the Raman shift the functional group of your polymers. Raman got able to get up to about 1 micrometer in size, whereas whereas FTIR could give you about 6 or 10 micrometers as the minimum spatial resolution at the moment for seeing microplastics. Are we doing something to harmonize different techniques? Yes, there is a group also in ISO that is called the Joint Working Group 1 that is dedicated to harmonize the different approaches for the pre-treatment and analysis of samples. So in conclusion, it's quite difficult to quantify and identify microplastics and nanoplastics from complex environmental samples with a single analytical method. Therefore, we might need a combination of multiple methods. On occasions, we want to have all the answers or as many answers as possible. Sampling methods for nanoplastics are still under development. Membranes with pore sizes down to 100 nanometers, or also continuous flow centrifugation has been recently reported for aqueous samples. Isolation or pretreatment protocols may be needed, for example, density separation, enzymatic digestion, H2O oxidation, or Fenton reagent. FTIR and micro-FTIR are one of the most common tools for the detection and quantification of microplastics. You don't need a visual pre-selection, particularly when using micro-FTIR, but you have a limitation of the minimum particle size, about 6 micrometers for micro-FTIR. Contamination, overestimation, or underestimation of microplastics in environmental samples may occur because of a lack of optimized sampling and analytical methods. However, reliable standards for the sampling and analysis of microplastics are now under discussion in the in ISO and in the different standardization bodies in different countries. So, well, thank you very much. These are some of the persons that have been crucial to part of the work that I have presented here, and the funding bodies and industrial collaborators that have helped us in many of the development of analytical techniques for the detection of these materials. And thank you very much all for listening.
Thank you so much, Jesus. That was amazing. I'd just like to bring everyone back. I think we'd like you to know a little bit. Can I ask that? But we've got so many great questions from the audience. I think we're just going to go through a couple of those, and we may run over slightly, but if anyone needs to leave, you can obviously watch back on the recording to get those questions as well. So I'll start with a question from Pabam. Tanya, you touched on biodegradable polymers a little bit, but to you and the others on the panel, what's your opinion of using biodegradable plastics? And is it possible or even desirable to replace all disposable plastics with either biodegradable plastics or other materials? And Tanya, do you want to start on that?
Yes, thank you very much for the question. It's a great question, and there was a lot of discussion in the EU, I know, with regard to, you know, should we look at biodegradable plastic as a solution. So I'll start by saying that while biodegradable plastic is great and it offers obviously great possibilities, it is not a silver bullet solution. And there is no point in trying, you know, to replace, say, PET bottles with biodegradable plastic, because PET bottles are great material. You know, all the properties that they have are amazing, and they're easily recyclable. Where biodegradable plastic would be a good solution is, you know, for applications where you could not prevent leakage of plastic into the environment. So, you know, we should change our behavior, be more careful about how we use plastic, but also then apply science, obviously. And for example, there are those trimmers that you use to cut the grass. And apparently they break very easily and they shatter into small pieces, and it's very hard to collect it. So using biodegradable plastic for application like that would be brilliant, but definitely not replacing all plastic with biodegradable plastic.
Did anyone else have anything to add to that question in particular? No, we've got another one here, which is more. Oh, sorry, Robin, were you coming in there?
No, it's okay. I was just going to say that also often biodegradable plastic is combined with conventional plastics still. So using a blend just means that you get those conventional plastics in smaller pieces more quickly.
Okay, so not an ideal solution at all in that case.
No.
Jesus, this question's more for you. It came from Ash and Aziz and a couple of others as well, actually. Are there simple ways for detecting microplastics and nanoplastics in water and food, potentially for citizens themselves or in places where they don't have as much scientific infrastructure as we are lucky to have in the UK?
Well, actually, it's not easy. As you can see, the of analyzing microplastics require a little bit of pretreatment because of the size. So that's also why it's so concerning, because we don't know what effect we might have with the microplastics, and the limited sizes in terms of microns or nanoplastics, we cannot see them easily. And if they are hidden in our food or in a specific sample, you might need to digest them, to pre-treat them. So for the analysis of microplastics in food, for example, what has been reported, or the analysis of microplastics in organisms such as worms or fish, you need to pre-treat the sample, you need to digest the organic matter, you might need to dissect your sample. So it's unfortunately not straightforward. Microplastics of 5 millimeters can be easier to see, but at the same time, it's not something that obvious. So it is indeed a challenge. Therefore, that's why it's so concerning.
Speaking of monitoring, Robin, do you know of any monitoring programs for plastosphere?
I'm not aware that there's any kind of, I mean, aside from often it's the same groups that kind of continuously do something fairly similar in a similar area. I don't think I've heard of anything that's like a consistent monitoring. I know I have heard of perhaps the potential for using things like the continuous plankton recorder samples to look for, you know, whether you can even realistically look at the plastosphere in those samples if plastics are collected. But as far as I'm aware, no one's done it.
Sorry to put you on the spot there. I was just aware we have a really international audience, so it'd be interesting to see if that could be linked up somehow to create some kind of monitoring scientific ongoing project. This is another question, thinking about internationally, this is for everyone, what is the best and simplest way of reducing plastic pollution in countries without a formal recycling system? And that came from Efulua and Kipkore. Anyone want to jump in to start? Tanya.
I suppose it's a great question, and you know, it's easy to talk about recycling targets and stuff like for the countries that have recycling in process. Think about what you need. We are creatures of habit, and the reason why I asked the question, like, you know, should we ban plastic, is you hear a lot of people saying, well, we should just not use plastic now. But the truth is that plastic makes our lives much easier, and it has some great positive effects. You know, you're using plastic to make, it's a lightweight material. You use it to make cars, car parts, and airplanes, which means that you can transport things, goods, to long distances and reduce carbon dioxide emission by doing so. So plastic is great. We need to use it. And where there is no recycling facilities, you know, think about the alternatives to plastic, or think about, do you really need another plastic item in your household? Because like at the top of the plastic waste pollution should be reduce and reuse, and then recycling comes as a third kind of option. So, you know, when you don't really need something, don't buy it. Toys, you know, it's great to just give them away when you don't need them. Clothes, same thing. So just think about reducing and reusing what you already have.
Anyone else want to add anything on that message? Jesus?
Yeah, I mean, that's absolutely true. I mean, possibly one of the most important things is education, and be aware of how I can reduce, how can I avoid the use of, for example, disposable or single-use plastics. Plastics are needed in many applications, but for packaging or a simple bottle of water, that is something that can be definitely changed. If one of the things that is striking is that the majority of microplastics in the environment come from secondary microplastics, which is the degradation of bigger plastics, and that is a problem of waste mismanagement, and it's just a matter of also be aware and be have some consciousness in where we put our rubbish and everything like that. Something that is very sad sometimes when there is a very nice day out of the beach or in a festival or in a concert. When you leave, there is plenty of plastic garbage on the soil, and that is something completely avoidable. So we can do better, taking a little bit more responsibility and education in dealing with this.
Fantastic, thank you. We've had so many amazing questions. I think I'm just going to take one last one. We've got lots of different ones for each of you, but I think I'm going to take one last one to round us off here. So this is about the costs associated with addressing the issues in terms of recycling, and mainly for Tanya. But so when we're talking about recycling plastic, taking plastic waste and using enzymes to recycle that and turn to high value products, what is the cost of that? Would you say that was more cost effective than other recycling methods? Is that part of the analysis that you do on these recycling methods?
It's not something that I do, but you can find the Turnier and colleagues, when they looked at the efficiency of recycling, enzymatic recycling of plastic, in that paper that I've referenced, they have actually done the cost of the process. So the point is that this process is not comparable to mechanical recycling. So mechanical recycling is still cheaper because for both processes you need to collect and you need to separate the waste. And in this particular case, this recycling is only, enzymatic degradation is only applicable to polyethylene terephthalate. So you have to really carefully separate it. You know, the process is economically not very comparable to mechanical recycling yet. But we're hoping to, by using engineering of enzymes, etc., we can create better processes and bring the costs to be more comparable. If we're talking about now, plugging in microorganisms, what I was talking about, we're not even close to talking about economics of the process. We need to develop so many other things, but that shouldn't stop us from trying. I think it's very important, because recycling does offer you an issue to a certain extent. So I did mention, we collect and recycle about 30%, only 4% goes back into the products. And the thing is, like, you can only repeat the process 2 or 3 or 4 times, and then the recycled material loses even more its properties and you cannot reuse it. So what you're going to do, you're going down downcycling route, and you're creating something of lower and lower and lower value. And inevitably you will use that plastic in, kind of weave for the carpets and things like that. And finally they will end up on the landfill. So what we're trying to do is do whatever we can to divert plastic from landfill. And that's what we're trying to offer as an option. It might not end up as a process that's going to be economically as close to recycling, but it's certainly going to be something that's going to be valuable, because plastic will not end up on the landfill.
I know I said that was the last question. I'm really briefly going to widen it out to Jesus and Robin. In terms of doing what we can, is there feasibility in taking plastic out of the ocean and removing it from that environment on a large scale, do you think?
I mean, I think it really depends. Like, I would say with current technology, no. I don't think that, I mean, things like the ocean cleanup, I think most scientists agree that it's a fairly problematic enterprise, because it's just, really non-specifically removing things from the ocean, and some of that is going to be plastics and some of it is going to be organisms. I don't really know what the solution is. I would say that there's not really anything that currently kind of fills that gap, from my perspective at least. I mean, Jesus, I don't know what you think as well, but
yeah, I mean, I agree. It's going to be very difficult to think, at least at the moment, of a strategy of collecting plastic pollution or microplastics in the ocean or in the environment in general. Also, something that happens is that not all plastics float, for example. So in the ocean, you will have some plastic that might be floating, like polyethylene or polypropylene, but some will sink, and you will have a big amount of plastic that are in the seabed at very deep meters, and that is already being possibly there picked up by other organisms in there, or simply staying there. It's going to be a mammoth task, and I think at the moment it's not going to be possible just to pick everything up. Plenty of things to rely on, like enzymatic digestion or bacteria, but nevertheless that takes time, and there is I don't think there is a simple answer at the moment to grab them all.
Okay, Tanya, very quickly.
Very quickly, just I don't want to finish on a very gloomy note. I don't think that we can solve all the aspects of the problem. We should try to make everything that we can to stop plastic getting into ocean. Good news is that bacteria are very evolvable, and they will easily find a way to use something that's in the nature. Don't forget that we've been using plastic, it's increasing the extent that we're using plastic over the last 50 years. Bacteria will definitely evolve to be able to degrade it. So, they will clean up the ocean, but what we need to do is stop spreading the waste. Thank you.
Thanks, Tanya, for that final message. Apologies again for running over, but we had an excellent discussion there, I think. And I want to thank our speakers once again for joining us today and giving your time to what has been a really interesting webinar. I'll just remind you quickly of the resources I mentioned at the start, especially the RSC explainers, which can be found on our website. And there's also some links there to the microbiology website if you want to have a look at some of their resources. There'll also be a recording of this webinar if you want to share it. And we've been running these webinars every few months as part of a series. So if you check back on the Chemistry World website for news about the next one, and sign up to that as well. Finally, I'd just like to say thank you to the Microbiology Society for joining forces to organise this excellent webinar. And that's all from me.
Thank you very much, Kathy. I obviously would like to thank you, Kathy, for joining us once again. An excellent host, not one of the RSC's journalists, but still does an absolutely fantastic job of hosting and moderating this sort of event. So thank you, Kathy. Look forward to the next one in a few months' time as well. And of course, a huge thank you to Rachel, who has done a very difficult job. Latin names for bacteria cannot be easy things to translate into British Sign Language. So thank you, Rachel. I hope everybody who needed Rachel's services there will agree that she did a fantastic job. So that is it for this time. So as Kathy was saying, at chemistryworld.com/webinars, you'll find out information about all the upcoming events. While you're there, if you register for a free account with Chemistry World, you'll get access to more stories, and you'll be able to sign up for our regular emails with the latest content and with information about things like upcoming events like this. So do please think about that while you're there. Keep an eye on your inbox over the course of the next few days for a link to the recorded version of this webinar and your certificate, which is our little way of saying thank you. So that is it for this time. Do please complete the survey on your way out. It really helps us to know what sorts of topics you want to cover, how well we're doing with our webinars in general. So please do take a couple of minutes to do that. We really appreciate it. And thank you for all of your excellent questions, for all of your fantastic engagement throughout this webinar series so far. So thank you once again to everybody involved. I'm Ben Valsler, Chemistry World's digital editor, and we will see you for the next Chemistry World webinar. Take care.